Welcome to today's webinar, First Annual Global Clinical Trials Update. Welcome from wherever you're joining us in the world. For this webinar, we have English and Spanish captions available. If you're joining us live on Zoom, please check the chat box as we're adding the instructions right now to access the captions. If you're watching the recording, you will find the instructions in the video description on how to activate the subtitles. Today, please use the question and answer section for your questions, the Q&A at the bottom of the screen. Just a caution, these presentations, some are live, some are recorded, and we may encounter some glitches with tech. Sometimes it doesn't work exactly as it should, and please be patient with our presenters as we work through anything unexpected. For those of you that don't know me, I'm Kathy Cummings, Executive Director of the International Alliance of ALS MND Associations. And today we'll be getting information on ALS MND treatments currently in phase 3 clinical trials. Today we're joined by AB Science, Amylyx, Biogen, Clene Nanomedicine, Cytokinetics, Ferrer International, Ionis Pharmaceuticals, and Mitsubishi Tanabe Pharma. Thank you all for joining us today to be able to give these updates to our ALS MND community. Our moderators today are Dr. Gethin Thomas and Dr. Thanuja Dharmadasa. Gethin has over 20 years experience as a biomedical researcher in musculoskeletal diseases, publishing more than 65 papers and securing close to $4 million in research funding. He has extensive reviewing experience, both as a manuscript reviewer and serving on Australian and international grant review panels. As a research manager, he's directed a university research office, higher degree research programs, and served as an associate dean of research. He has extensive experience on high-level research strategy, as well as a deep understanding of the grant system for the preparation, application, and review process through to management of funded projects. Dr. Thanuja Dharmadasa is a neurologist and senior clinical research fellow at the Royal Melbourne Hospital and the Florey Institute of Neuroscience and Mental Health in Victoria, Australia. She graduated from medicine with first-class honors and completed her neurology specialist medical training in Victoria, undergoing a clinical neurophysiology fellowship in her final year. Her PhD in MND was conducted at the University of Sydney, Australia, and during which time she was awarded the Jim Lance Young Investigators Award and the Peter Bancroft Prize for her work. Following this, Thanuja received a prestigious NHMRC C.J. Martin Early Career Fellowship to continue her work at the University of Oxford, UK, as a postdoctoral clinical research fellow. Her research explores novel clinical and advanced neurophysiological and imaging techniques in patients to understand disease mechanisms in MND and to uncover disease biomarkers. She has worked as an MND clinician at several MND specialist care centers, both nationally and internationally, and has a keen interest on strategies to optimize best care integration of research for patients living with the disease. Thank you for moderating today's webinar, Dr. Dharmadasa and Dr. Thomas. I will now turn it over to the two of you to moderate for today.
Thank you, Kathy. And I'd just like to thank all the people dialing in today for listening to this presentation, and also thanks to all our presenters. Thank you. That's on behalf of myself and Thanuja. We'll be rolling through this pretty quick today. We've got a series of presentations, and after each presentation, we'll have time for a couple of very brief questions, really relating specifically to those presentations. Then after all the presentations are finished, we will have a panel Q&A where we can dive into any of the questions a bit more deeply. So just to get this moving, I said we were on a very tight schedule. So our first presentation is from AB Science, talking about their studies on masitinib, and we'll have Christian Fazot present for the Q&A after the session. Thank you.
Good morning. My name is Christian Fassot. I am the Chief Medical Officer of AB Science, and today I'm going to talk to you about masitinib, a potential breakthrough therapy in ALS, which is currently in conditional registration process, but also about its late-stage development in progressive multiple sclerosis and Alzheimer's disease. The use of masitinib in ALS is supported by a well-documented and differentiating mode of action, which is based on neuroprotection at both central nervous system and peripheral nervous system levels. Masitinib exerts a protective effect at the central nervous system via microglia and at peripheral nervous system via mast cells. You have here several items related to in vitro and in vivo animal models demonstrations which have been published quite recently, several times from 2016 to 2021. The development program of masitinib in ALS is supported by one completed phase 2b/3 study, which has been published regarding its main results, but also regarding its long-term follow-up, respectively in 2020 and in 2021, and also a second phase 3 study, which is currently ongoing. Both studies based on a long time endpoint of 48 weeks. So the first study, AB 10015, demonstrated a significant delay of disease progression over 48-week period in the primary analysis population of the normal progressors with masitinib 4.5 milligram. As you can see in this normal progressor subpopulation, which was the primary endpoint, 27% slowing of functional deterioration has been shown. Important to note is that these results remain positive when imputing in sensitivity analysis all the missing data, both through multiple imputation model and multiple imputation with penalty. When we add the fast progressors to the normal, which was a sequence of analysis foreseen, we lose significance, and results become not significant. To be noted is that the 3 milligram per kilo per day is also not statistically significant. The functional benefit on the functional score, ALSFRS revised, was supported by improvement in secondary endpoints with quality of life, 28% improvement in quality of life on items that really are making a difference for patients, 22% improvement in respiratory function, and 25% delay in disease progression, PFS, which is a time to event related to disease progression or death, plus 4 months. So we also perform a long-term follow-up of the patients from this study during 75 months from diagnosis, and this post-hoc analysis showed an increase in overall survival of 25 months in the moderate ALS patient, defined at least 2 on the ALSFRS items for the 4.5 masitinib arm compared to control. Important to note is that this 25 months or 44% reduction risk of death through an overall survival sensitivity analysis was already present at the end of the blinded period of the study with a reduced risk of death of 65%. Based on those results, a second phase 3 study has been initiated to further evaluate masitinib in the moderate ALS patients, and also to test a higher dose of masitinib, namely the 6 milligram per kilo per day dose. The study intends to include the patient with 24 months disease duration, moderate ALS patients that I mentioned, and we exclude the slow progressors in this study, including 10%, 45 patients of fast progressors in order to perform exploratory analysis. The study is currently ongoing and intends to recruit almost 500 patients over 48 weeks. You have the list of approved countries on the right. If we move away from ALS but remain in neurodegenerative diseases, we are also engaged in phase 3 in progressive multiple sclerosis. This study is currently enrolling patients and intends to enroll 800 patients over a 96 weeks period. The study is based on results generated on a completed phase 2b/3 study in primary progressive MS, as well as non-active secondary progressive MS, which enrolls 600 patients over 96 weeks and show positive results on primary endpoint with a significant reduction in progression of ATSS, and also on secondary endpoint with a 42% risk reduction of time to disability progression. The last indication in neurodegenerative diseases is Alzheimer. There as well, we are starting a phase 3 study, which is going to enroll 600 patients over 24 weeks duration. This study is based on results generated in a completed phase 2b/3 study, which enrolled over 700 patients in 24 weeks duration, and showed a positive significant effect on both primary endpoints of ADAS-Cog and ADCS ADL. So this brings me to the end of my presentation. I thank you for your attention. Thank you very much.
Thank you for that. We've just got time for one, I think, very quick question since we had a bit of delay there. The doses used was 4.5 milligrams the highest dose you used in the MND trial, and is the same dose being used in your MS and Alzheimer's trials? Can I just ask that?
So in the ALS trial, in the phase 2b/3, we used 3 milligram per kilo per day and 4.5 milligram per kilo per day. In the MS study, we used 4.5 and 6 milligram per kilo per day. And in Alzheimer, the same. So in the confirmatory phase 3, we use in ALS 4.5 milligram per kilo per day, and we are testing the higher doses of 6 milligram, which we know from other indications, in order to see if there is an additional potential benefit in terms of efficacy at let's say, reasonable cost in terms of safety. And for the two Alzheimer and MS phase 3 studies, we are using the 4.5 milligram.
Thank you, Christian. We'll move on. Thanuja?
Yeah, so our next presenter is Michelle Manuel, and she's the Global Head of Medical Affairs at Amylyx Pharmaceuticals. She'll be giving us a clinical trials update of Amylyx in US and Europe.
Hello, I'm Michelle Manuel, Global Head of Medical Affairs at Amylyx Pharmaceuticals. It is my pleasure to bring you an update on the ongoing Phoenix phase 3 clinical trial. The oral co-formulation of sodium phenylbutyrate, also known as PB, and taurursodiol, or TURSO, also known as ursodoxycultarine, developed by Amylyx Pharmaceuticals, is now commercially available in Canada and the US. This combination is also known as AMX0035, but for this presentation, I will use PB and Terso. Outside of the US and Canada, PB and Terso is an investigational drug and not approved for use. And currently, the review is ongoing by the European Medicines Agency. The key inclusion and exclusion criteria for Phoenix include that participants have clinically definite or clinically probable ALS, as defined by the revised El-Escorial criteria, be less than 24 months from symptom onset, have a slow vital capacity of greater than 55%. In the 48-week randomized placebo-controlled phase of Phoenix, assessments will be made of function by ALS-FRSR and survival. Lung function will be assessed with slow vital capacity. Participant quality of life, as well as caregiver burden, will be assessed using surveys. And of course, important safety measures will be assessed throughout the trial. The trial is underway. Recruitment is completed in the US and is ongoing in Europe. Shown here are the countries with enrolling sites. Now that PB and Terso is commercially available in the US, if a US Phoenix participant and their clinician decide to continue treatment with PB and Terso, they will transition to commercial product. US trial investigators have been notified about specific processes. Phoenix participants in Europe completing their 48-week randomized phase will be eligible to enroll in an open label extension, or OLE, phase and receive PB and Terso for up to 2 years. The Phoenix OLE will not open in the United States. US participants will, however, be followed for long-term survival analysis. In conclusion, Phoenix is underway and recruiting in Europe as planned. Phoenix features a participant-centric design and is designed to be telemedicine friendly with a total of 6 in-person visits over 48 weeks and remote assessment of both ALS-FRSR and slow vital capacity. The additional OLE phase will further extend the assessment of safety and efficacy of PB and Terso in ALS. For additional information on the Phoenix trial, visit www.clinicaltrials.gov and search for NCT 05021536 or check out www.amylyxalstrial.com. Thank you for your attention, and a special thank you to all of the Amylyx trial participants.
Well, thank you very much, Michelle, for that update. And there's undoubtedly going to be lots of points of discussion, particularly given the recent approval by the FDA. But I guess if I can ask one quick question, you mentioned that the phase 3 trial in Europe is ongoing. Is the overall thought that if that turns out to be negative or it doesn't reach its endpoints, well, what's the position of Amylyx in regards to continue that drug in the US?
Yeah, thank you for that question. And I think if any of you watched the FDA advisory committee, we very publicly said if the trial is absolutely negative and does not show benefit, we're willing to consider, and working with the FDA and with the Canadian health authorities as well, to do what's right for people living with ALS and for people taking Relyvrio in the US or Albrioza in Canada, including up to the drastic measure of withdrawing the product from the market if that's what it takes. But we're confident in our study investigators, and we will let the data lead us.
Great, thank you. So probably we'll move on to the next presentation.
Thank you, Thanuja. Our next presentation is from Biogen. We're talking about the long-term evaluation of BIP067, otherwise known as tofersen. And we'll have Stephanie Fredette, Senior Medical Director, available to answer the Q&A afterwards. Thank you.
Hello, everyone. It's a pleasure to be with you virtually today. My name is Stephanie Fredette, and I'm a Senior Medical Director at Biogen. In my role, I oversee clinical development for our ALS portfolio, including tofersen, which I'll speak about today. Next slide, please. Tofersen is an investigational drug being developed for the treatment of ALS associated with mutations in the superoxide dismutase 1 gene, or SOD1 ALS. In people living with SOD1 ALS, which represents about 2% of the ALS population, mutations in the SOD1 gene lead to production of a toxic form of SOD1 protein. Tofersen is an antisense oligonucleotide, or an ASO, designed to target SOD1 messenger RNA in an effort to reduce the creation of SOD1 protein. Tofersen has been evaluated in several clinical studies, 2 of which are ongoing, and I'll speak about today. And a marketing application for tofersen is currently under review by the US FDA. Next slide, please. The first of the ongoing studies is the open label extension study 233-AS102, which was designed to evaluate the long-term effects of tofersen in people who previously completed the parent study, or study 233-AS101. Enrollment in this open label extension is complete, and the study is expected to conclude in 2024. As you may be familiar, combined results from the pivotal phase 3 VALOR study and an interim data cut of this OLE were published in the New England Journal of Medicine in September of this year. And these data help us understand the effects of tofersen in the symptomatic population. Next slide, please. The other ongoing tofersen study is the ATLAS study, or study 233-AS303. ATLAS was designed to evaluate whether pre-symptomatic initiation of tofersen can halt or delay emergence of clinically manifest ALS and or slow the progression of disease. ATLAS is a 4-part study, and I'll walk through each of these parts briefly. Part A is a natural history run-in where pre-symptomatic SOD1 mutation carriers undergo monthly blood draws to monitor for elevations in neurofilament. Neurofilament is a protein that is released from neurons when they're degenerating. And in a separate natural history study called PREFALS, led by Dr. Michael Benatar and his team at the University of Miami, neurofilament levels were found to increase prior to emergence of signs or symptoms of ALS in at-risk mutation carriers. Participants who meet the protocol-defined neurofilament threshold in Part A will be screened for Part B. In Part B, participants are randomized 1-to-1 to receive tofersen or placebo for up to 2 years, or until they develop clinically manifest ALS or signs and symptoms of ALS, at which point they would have the opportunity to receive open label tofersen in Part C. And finally, for participants who have onset of signs and symptoms of ALS in Part A, before they reach that neurofilament threshold, they may be eligible to receive open label tofersen in Part D of the study. Next slide, please. The ATLAS study is enrolling clinically pre-symptomatic adult carriers of highly penetrant and rapidly progressive SOD1 variants who have plasma neurofilament levels that are below the protocol-defined threshold, or 45 picograms per mil. It's important to note that this threshold is specific to the assay being used in the ATLAS study. The protocol includes a predefined list of SOD1 mutations, which are shown on this slide. And these mutations are well characterized to be highly penetrant and associated with rapid disease progression. For other SOD1 variants that are not listed, there is an independent mutation adjudication committee in place to determine individual variant eligibility based on literature, family pedigree, and other sources. Excuse me, next slide. The ATLAS study aims to enroll 150 people in the natural history run-in to ultimately enroll 28 of those 150 in the interventional Part B of the study. We're currently enrolling at 29 sites across 14 countries shown here, and any individuals who are interested in participating should speak with their doctors to obtain more information. Next slide. I'd like to close just by sharing my immense gratitude for all of our participants across all of our ALS trials and their families and their caregivers, because we could not do this without you. Thank you.
Thank you for that. Just a quick question before we move on to the next talk. How long is the readout going to be, or how long until we get the readout from the ATLAS trial? I might have missed that in the in your presentation.
Thank you for the question. Hello, everyone. So the readout will depend on how quickly we enroll part A and then ultimately part B of the study, but we're anticipating it as early as 2026, 2027 timeframe.
Will there be any opportunity to sort of look at the data, have a sort of interim view of the data to see if there is any strong effects? Because obviously it's a really interesting design of the study and really unique. So we're all very excited to see what happens with it.
Yes, we share that excitement. So we are contemplating a potential interim efficacy analysis, and a lot of that will depend again on the time of enrollment and how individuals are progressing through the different stages of the study. So that is a potential aspect of the protocol. If we were to conduct an interim efficacy analysis, again, depending on timing of enrollment, it may pull timelines in ever so slightly to 2025, but not in the near term.
Thank you, Stephanie. We'll move on to our next presentation now.
Yep, so up next is Rob Etherington. So he's the CEO and president of Clene Nanomedicine. He'll give us an update on the Healy ALS platform trial.
My name is Rob Etherington. It's a pleasure to talk to you today. I'm the CEO and president of Clene Nanomedicine, a clinical stage biopharmaceutical company focused on revolutionizing the treatment of neurodegenerative disease because of how our lead asset, CNM-Au8, targets energetic failure. Specifically, what our drug is doing is driving the gas, the fuel that your central nervous system, your brain and mind relies upon for energy, which is to say nicotinamide adenine dinucleotide and adenosine triphosphate. By targeting energy metabolism, our thesis, and we've seen this in preclinical animal models as well as in the clinic, as you're about to see, is to improve energy production and its utilization within the central nervous system. This is a critical market need. Over the course of the last number of years, we've been building the clinical case for neuroprotection and remyelination. As you see on the far right, in Parkinson's and MS subjects, we demonstrated target engagement, which is to say patients drinking our drug by mouth had it go to the gut, through the bloodstream, back up to the head, into the brain. On the far right, we presented data in August for the visionary multiple sclerosis study that showed neurological improvements in people with stable relapsing MS as adjunctive therapy or on top of standard of care disease-modifying therapy drugs. Now, let me focus most importantly on ALS. We released rescue data last year and Healy data only this last month, early October. And I'll review this data that is very relevant to you and I. The rescue study, though it missed the primary endpoint, we saw a most interesting, even important survival benefit occurring by following these patients over time. The green line represents all the actives during the 2.5+ years, 2.5 years plus a follow-up period in the open label extension. The gray dotted line represents the placebo. As you see here, we have achieved or demonstrated a 70% decreased risk of death with a hazard ratio of 0.29 and a p-value of 0.01 over these 2.5 years of follow-up. We were very gratified and found it also important that in the Healy study at 6 months, CNM-Au8 30 milligrams, the same dose in rescue, demonstrated a more than 90% risk reduction of death, no matter how we looked at this with respect to either the efficacy regimen or the full analysis set, including all placebos across the regimens, for either death or permanently assisted ventilation or death alone. Now, only the death or PAV two groups were statistically significant in the sense that they achieved in the adjusted Cox proportional hazard ratio a statistical significance less than 0.05, but we see a most interesting signal also achieved in death alone. So what's next? We continue to follow these patients in rescue because we haven't even neared what is termed median survival, which will answer rather the question, how long are these patients able to live from this smaller rescue study? And we will yet see the open label data from the Healy program, as well as the last patient, last visit data that will emerge from the Harvard Healy clinical database over the coming months. Clene will continue supplying drug in open label extension to all of these patients in both programs. In addition, we continue to supply expanded access protocol CNM-Au8 through Protocol 1, which was FDA approved, and now through another Protocol 2 in an expansion for patients that have requested it from other clinical programs where they have not achieved success. Our next step is to meet with regulatory authorities and to commence a global phase 3 study to evaluate the survival signal and to prove it in larger numbers. Certainly, we recognize that we need time under our belt for CNM-Au8 to drive the mortality or survival benefit that we're seeing in these programs. And in the meantime, Clene is committed to continuing the ability for patients in these protocols to access our asset. Thank you very much for your attention. If you have further questions, we look forward to conversing with you. You can reach out to info@clene.com, where you will be able to connect to our medical affairs team. Thank you for the time and attention. Let us all fight together in solving this devastating condition we call ALS.
Well, thank you, Rob. And again, some very interesting data and points. I guess just to ask you one question, start with, you mentioned the survival benefit in the rescue ALS trial at the 30 milligram dose. Was this, you might have mentioned this, but was this result limited to that dose, or did you also see it in other doses that was trialed in that study? I think you might be on mute.
Forgive me. The rescue study only evaluated 30 milligrams versus placebo. It was our sole dose that we evaluated in that study. When we went to the Harvard Healey program, the agency asked us to look at a dose response, and we doubled 30 milligram to 60, and we did not see any survival signal in 60 in Healey. So we have concordance of 30 milligrams between both studies.
And were there any kind of safety-related differences or concerns that might have
There was not. Yeah, there was not. In fact, we don't have a single serious adverse event that has been related to drug in any of our clinical programs across ALS or MS to date, for either of 30 or 60, in fact.
Thank you. Then we'll move on to the next talk.
Okay, next presentation is from Cytokinetics, talking about their courage and vitality trials. And we'll have Johan Brecker, Medical Director from Cytokinetics, available for the Q&A. Thank you.
Greetings, my name is Tyrell Simpkins. I am an Associate Medical Director at Cytokinetics, and I am going to be giving you a brief overview of our phase 3 trial termed Courage ALS of reldesemtiv in people with ALS. First, I'd like to discuss the study design of Courage ALS. Reldesemtiv is a fast skeletal muscle troponin activator and is being investigated as a potential therapy to slow the decline of skeletal muscle function in patients with ALS. In our phase 2 trial of reldesemtiv, Fortitude ALS, participants within 24 months of diagnosis were assigned one of 3 doses of reldesemtiv. Our primary endpoint, which was the change in the vital capacity, was not met, but in all doses, reduced progression compared to placebo. And this included the ALS decline in the ALS FRSR, vital capacity, and strength. Combining all doses, we found that a decline in the ALS FRSR was statistically significantly reduced in reldesemtiv treated patients. This promising result led to our pursuit of Courage ALS, which is our phase 3 multi-center double-blind randomized placebo-controlled trial with a goal to evaluate the efficacy and safety of reldesemtiv in patients with ALS. Presented here is the overall study schematic of Courage ALS. It is a global study in 16 countries with 85 sites. Key inclusion criteria include an FVC greater than or equal to 65% predicted, symptoms onset of less than or 24 months, and an ALS-FRS-R equal to or less than 44. We will be screening 555 patients, and randomization will be 2-to-1 reldesemtiv versus placebo. The first 24 weeks will be a double-blind portion, after which all participants will enter an open-label phase and will be receiving reldesemtiv. The primary endpoint in this study is at the 24-week mark and is the change in the ALS-FRS-R with secondary endpoints of the FVC, LSAT Q40, and muscle strength. As you can see in the bottom, there are a number of visits, approximately every month, but you will notice that there are different colors between in-clinic and remote visits built in for intentional flexibility as well as continued monitoring. The in-clinic visits are day 1, week 12, week 24, week 36, and then finally week 48, with remote visits in between those. Presented here is a brief overview of the distribution of Courage ALS sites throughout the world. We have 36 sites in the US and 11 in Canada, 5 in the US, and then the remainder are sprinkled throughout Europe at multiple different sites. As mentioned before, the primary endpoint in Courage ALS is the change in the ALS-FRS-R at week 24. Secondary endpoints include change in the forced vital capacity, hand grip strength, and LSAT Q40 at the week 24 mark. And then there are a number of exploratory outcomes, including the usage and prescription of durable medical equipment, handheld dynamometry, the EQ-5D5L, as well as the visual analog score, and then time to first hospitalization. There are a number of patient-friendly characteristics that we have intentionally built into Courage ALS that I would like to point out. First of all, non-invasive ventilation at screening is not an exclusion to entry into Courage ALS. More than half of the trial visits are remote, and there is intentional flexibility even in the in-clinic visits to accommodate the various needs of patients with ALS. Randomization is 2-to-1 favoring active treatment, and the 24 weeks of placebo-controlled treatment is followed by 24 weeks of open-label active treatment for all participants. Cytokinetics is also committed to continued treatment with reldesemtiv for participants who complete 48 weeks. And we can say that definitively as we have already opened up and began enrolling in the Courage ALS open-label extension. This open-label extension will go for another 48 weeks for those who have participated and completed Courage ALS, with, again, intentional flexibility in both in-clinic and remote visits. The primary objective of the Courage ALS open-label extension is to assess the long-term safety and tolerability of reldesemtiv in patients with ALS, with a secondary objective to assess the long-term effect of reldesemtiv on ALS, FRS-R functional outcomes and hospitalizations by comparing early start to delayed start groups from participants in Courage ALS. The assessments in the open label extension include the ALS-FRS-R, continued monitoring of durable medical equipment use and hospitalizations, and then there will be a number of safety assessments including examinations, adverse events, evaluations, and then clinical safety labs. Lastly, I'd like to also present the ongoing commitment of Cytokinetics to people with ALS, including the reldesemtiv Managed Access Program. We are preparing a managed access program for patients that previously received either tirasemtiv, our drug prior to reldesemtiv, or reldesemtiv. At the end of 48 weeks of dosing in the open label extension, patients may then transition to a reldesemtiv Managed Access Program. If the treating physician agrees to participate in the program, the treating physician's patients may be eligible to transition from the open label extension to the Managed Access Program. We at Cytokinetics would like to particularly thank all of those people with ALS that have participated in our studies with our mutual goal to find new and effective therapies to combat this devastating disease. I thank you for your time and attention.
Thank you for that. Shay, can you just turn my video back on? I think you turned it off and I can't get it back on now. Cool. All right, thank you for that. Johan, one quick question. It's really fantastic to see yourselves and other companies are developing these more inclusive, flexible trials. Have you found that it's easier to or faster to recruit into your trials? And also, do your patients tend to sort of finish the trial and then you get a better recruitment into the OLE as well?
That's an excellent question. That's an excellent question. Thank you for having me. I think the proof will be in the pudding in terms of seeing in hindsight how these criteria that are more friendly towards participation actually end up affecting clinical trial enrollment. But it is the hope that with some consultants in the patient community and taking their advice in designing these studies, that enrollment will hopefully accelerate. So I think to be determined, but that is definitely our hope.
Thank you. Over to you, Thanuja.
Great. So for our next talk, we'll be hearing from Lubia Alvarez, who's the Clinical Development Medical Lead at Ferrer, to discuss the ADORE oral edaravone trial.
Hello, my name is Lubia Alvarez. I am Clinical Development Medical Lead at Ferrer. I'm part of the ALS team here at Ferrer, and we are very pleased to be here. Thank you very much for the invitation. I'm going to talk to you about the ADORE study. ADORE is an international phase 3 randomized placebo-controlled trial with daily edaravone in ALS. So we are researching the safety and efficacy of FAB-122, this is our IMP, edaravone, for people living with ALS. FAB-122 is a newly developed oral formulation of edaravone that has to be self-administered daily. This compound can help to prevent oxidative stress, which we know is one of the causes of self-nerve in ALS. And this could potentially protect motor neurons and slow ALS progression. Participants will be randomly assigned to receive either edaravone or placebo in a ratio 2:1. That means that from every 3 patients, 2 will receive edaravone and 1 will receive placebo. Patients are allowed to use as a background therapy riluzole in doses of 100 milligrams a day or less. And both placebo and edaravone will need to be dissolved in water and ingested orally daily as a drink. This is the timeline of the study. We are planning to enroll 300 patients living with ALS. And when they start the trial, they will receive either edaravone or placebo for 48 weeks. The study started in October of 2021, and it is estimated to be completed by Q2 of 2024. Currently, we have 35 sites active enrolling patients. We have several trial centers all across Europe participating in this trial. And if you want to find your participating center the closest to you, you can access trials.org to look for this information. It is very important for Ferrer to provide the patients with the option of this treatment. So after 48 of treatment in ADORE study, all participants will be offered the possibility to roll over in an open extension study to receive daily oral edaravone until the drug is available in the market. Besides this open label extension, in the patients participating in the ADORE trial, the patient's expenses are covered. This means transportation, meals, or accommodation if it's necessary. If you want to have more information about the trial, we have an open website through Ferrer Medical Information, or you can also contact info@trials.org. Thank you very much.
Thank you very much for that presentation. And I guess this has been quite highly anticipated since the IV edaravone trial and the approval there. I just want to ask a quick question about your inclusion criteria. Given that the IV edaravone benefits of the drug seem to be in a very particular set of patients who had early clinical disease, did that inform your inclusion criteria for this study, or are you doing more of a wider generalized study to start with? I think you might be on mute.
Do you hear me now?
Yeah.
Yeah. Okay. Thank you. Sorry. So yes, we're trying to include as much patients as possible. So yes, our criteria is much more wider. So we're planning to go for a profile of subjects with moderate disease, and the inclusion criteria are basically working towards that. So we can have access, and the patients can have access to the trial in most of the population of ALS patients.
And have you seen from anything, it might be too early to say, but has it reflected what has been seen in the previous intravenous version, or is this looking like it's a different type of readout?
We haven't seen anything since it's a blind trial, nothing definite. We are performing all the safety follow-up and so on. But in terms of efficacy, there's no data yet available.
Great. So we'll move on to the next talk.
Okay. Thank you, Thanuja. Our next talk is from Ionis Pharmaceuticals, who are going to talk us about their studies on Ion 363, which is the FUS genetic therapy. And we'll have Danielle Edwards and Chris Yun available to respond to your questions. And please do ask your questions. Fire them in on the Q&A or even the chat. We're keen to hear what the audience want to know. So thank you. Please take it away.
Hello, good afternoon. I'm Becky Crean, Executive Director of Clinical Development at Ionis Pharmaceuticals. I want to first thank the Alliance for inviting me to provide an update on our ALS program. We've made some advances since we last provided an update, so I'm happy to share those with you. So as background, Ionis was founded about 30 years ago with a single mission. We wanted to turn antisense oligonucleotides, or ASOs as we call them, into a therapy. So ASOs are synthetic strands of DNA or RNA, and they bind to specific sites on the RNA to correct something that's missing or coded incorrectly. So the ASO we've developed for the treatment of ALS focuses on the mutation of the fused in sarcoma gene, or FUS gene. And so what is the biology behind FUS? And how does our ASO work to treat it? To better understand ASOs, you have to go a little bit back to your high school biology days and to the central dogma of biology. So this is just an oversimplified explanation of this concept, but basically DNA makes RNA and RNA makes protein. So if you see on the left, the DNA contains the information needed to make all of our proteins. It's the material that codes all of our genetic information. So the DNA is like the recipe book of life, and they are instructions that live within our cells that tell our body how to produce every protein that we need. Now that said, some proteins are harmful, and when our body's not producing some types of proteins correctly, those can be harmful as well. So in FUS ALS, there's a change in the DNA, and the information in this DNA is converted into small, portable, single-stranded RNA messages. And in FUS ALS, this RNA copy is a copy of the DNA that carries this abnormal code. And so this means that the information needed to build the fused in sarcoma protein is not correct. So you have this abnormal code in the RNA telling it to make more FUS protein than our body needs, causing the FUS protein to be toxic to our bodies. And it's this overproduction of the FUS protein that is thought to cause this specific form of ALS. So you can interrupt that process or fix that with an ASO. So you see kind of on the bottom half of the screen, an ASO is a small string of nucleotides that you can design so that it binds specifically to the RNA of interest, which you see in the middle panel. Then they either activate the protein production or they can also deactivate protein production. But in FUS ALS, the ASO deactivates the protein production so the body doesn't overproduce it. So we've taken this ASO called ION 363 and initiated a phase 3 global pivotal registration trial to generate and collect the information needed, hopefully for regulators to eventually approve the drug so that all people living with FUS ALS will have access to it. We initiated the study last year and will continue enrolling for the double-blind placebo-controlled portion of this trial for probably the next couple of years, as it takes some time to enroll a study like this. And that's the top orange bar that you see there. So once participants complete the first part, which is 18 months of treatment, they'll roll into an open label extension. That's in the middle orange panel. And this OLE extension will last anywhere from 2 to 5 years. We do plan to keep this open so participants can have access to the drug until we have marketing approval. So in the meantime, which you see in the bottom panel, in the meantime, we continue to work our way around the globe and work with the regulatory agencies so that we can open sites across the broad geographical area. So you can see we'll be doing this well into 2025 and likely some years beyond that as well. So where is this study taking place? Well, it's actually taking place all over the globe. We've got a number of sites in the United States that are active and enrolling. We also have sites in Canada, the UK, Netherlands, and Belgium that are all active. We're also in the process of activating sites in South America, other parts of Europe you see in the middle of the screen, such as Ireland, Sweden, Spain, Italy, France, Switzerland, Poland as well. And then we're also planning on opening sites in South Korea and Japan. So even though we plan to have about 23 sites spread out in over 16 countries, the closest site may still require some travel to get to it. So we've set up a travel assistance program through a company called Clene to help with getting to a site. This particular trial, the Fusion trial, covers all study-related costs for the participant and their study partner when they need to go to the study clinic. But also this group called Clene, they'll actually help you plan and book all of the travel as well. So they'll help you plan an itinerary and book airfare, trains, hotels, everything associated with travel. So we hope this program helps keep the focus on the treatment and the study participation and not have to worry about the travel arrangements and paying for being in the study. So in addition to our travel program, we're also providing no-cost genetic counseling and testing through our sponsored genetic testing program. So the testing program includes all ages to be eligible. Your doctor just must suspect ALS or you have a family history of ALS. So for individuals under 18 years of age, you have to have symptoms in order to be tested. But for individuals above 18 years of age, you can be pre-symptomatic, meaning you don't have symptoms, but you have a family history of ALS. So in addition to the testing, we're also providing genetic counseling through a company called Gene Matters. So what they do is they provide pre and post genetic counseling. So that means prior to getting a genetic test, and then they'll also provide genetic counseling after you receive the results of the test. So the genetic test requires blood, saliva, or a buccal swab specimen, which is collected from the potential participant. It gets sent to the lab for testing. We have a panel that tests 33 different genes on it, so it covers quite a broad range. To find out more about this program and how to access it, you can go to the website on the bottom of the screen for Prevention Genetics, and it gives you some more information about it. So I'll end by providing some information here about how to get in contact with us at Ionis and to learn more about the Fusion trial. There's a direct phone line listed here and an email address to get more information. You can also access the Prevention Genetics website too and learn more about our genetic testing. So we hope that you do reach out. So with that, I want to express my sincere appreciation for your time today and for listening to our update. And all of us at Ionis are deeply committed to continuing our work on behalf of the ALS community. And we can't do this without you. So we want to thank you for your commitment to doing this together. Thank you.
Thank you for that. Just time for a quick question now. I might have missed it, but how often is the injection, and is it intrathecal injections as with other ASOs?
Yes, thanks for the question, Dr. Thomas. Yes, it's an ASO. It's an intrathecal injection. The maintenance dose is every 3 months, but there's a loading dose at 1 month. So day 1 month, then 2 months later, then from there on every 3 months.
Okay, thank you. So we'll just move on quickly and then we'll come back for questions as long as they keep coming in. Our last presentation, over to you, Thanuja.
Yeah, so our last speaker today will be Alejandro Sala from Mitsubishi Tanabe Pharma, and he will share some highlights of their work, particularly discussing IV and oral edaravone for ALS.
Hello, my name is Alejandro Sala, and I'm the Medical Director in Medical Affairs for ALS at Mitsubishi Tanabe Pharma America. On behalf of Mitsubishi Tanabe Pharma, I would like to thank the conference organizers for inviting us. It is my pleasure to speak with you today about the work we are doing to help address the great unmet needs for patients with ALS. Mitsubishi Tanabe Pharma Corporation has been developing breakthrough medications for the past 3 centuries, starting in Japan, but growing to several countries around the world. Here in the US, we have an established medical affairs, sales, and marketing organization, Mitsubishi Tanabe Pharma America, a clinical development organization, Mitsubishi Tanabe Pharma Development America, which obtained FDA approval of both IV Radicava and oral Radicava ORS, and a management organization, Mitsubishi Tanabe Pharma Holdings America. We are committed to helping patients with ALS, and the first drug we launched here in the US in 2017 was Radicava edaravone IV. And just this year, we also launched the oral formulation of edaravone Radicava ORS. I would like to share with you some highlights of our clinical development program, along with recent publications that have helped to advance our knowledge and understanding of ALS. For Radicava IV, there were 2 phase 3 studies conducted, known as Study 16 and Study 19. Based on the results of Study 19 as the pivotal study, the FDA approved Radicava IV for patients with ALS in 2017. The post-hoc analysis listed here explore a variety of aspects of the effects of Radicava IV in patients with ALS. Most of these were focused on the generalizability of the effects of Radicava in patients with ALS. That includes the articles published by Scheffner and others in 2020, Al-Chalabi and others in 2021, and 3 articles published by Brooks and others this year. Here is one of the post-hoc analyses of Study 19 that was listed on the previous slide. This analyzed the long-term efficacy of IV edaravone in patients with ALS. The article was published in the journal Muscle and Nerve late last year. Here is another post-hoc analysis of Study 19, which was published this year. The article is entitled Edaravone Efficacy in ALS with Reduced Forced Vital Capacity, Post-hoc Analysis of Study 19. In October of this year, we published an article in the Lancet affiliate journal eClinical Medicine entitled Intravenous Edaravone Treatment in ALS and Survival, an exploratory retrospective administrative claims analysis. This was an evaluation of the overall survival in US patients with ALS treated with IV edaravone in a real-world setting. As you can see, Study 19 has been a rich source of data for us, and we are currently writing another article on a postdoc analysis of that study that analyzes the rates of hospitalization, tracheostomy, ventilation, and death in patients with ALS. As part of our commitment to patients with ALS, we have developed an oral formulation of edaravone Radicava ORS. To date, we have conducted 7 phase 1 clinical pharmacology studies on Radicava ORS in healthy volunteers and in patients with ALS with and without PEG or nasogastric tube, as shown in this slide. 5 of these studies are already published, and the 2 remaining studies will be published soon. The results of these studies have been presented at several national and international congresses, including NEALS, MNDA, NCALS, MDA, AAN annual meetings, among others. Our Radicava ORS development program also includes 4 phase 3 studies as shown in this slide. These are multinational clinical trials with study sites in the US, Canada, France, Germany, Italy, South Korea, and Japan. We have completed the AO1 safety study, and its extension study AO3 is ongoing. Studies AO2 and AO4 are also ongoing, and the objective of this is to compare a daily dosing versus our current regimen of Radicava. Results from these studies have been also presented at a number of medical congresses both in the US and internationally. As a result of these clinical pharmacology studies and the clinical safety study AO1, Radicava ORS was approved by the FDA in May of this year and is now available for patients with ALS. I would like to mention also that as part of our clinical program for Radicava IV and Radicava ORS, we are conducting the Radicava edaravone findings in biomarkers from ALS, the REFINE study. As some of you may know, a diverse set of biomarkers are proposed in ALS, but few have been validated, and this is an important area of active research. The primary objective of REFINE ALS is to identify putative biomarkers to serve as quantifiable biological non-clinical measures of edaravone's pharmacodynamic effect in ALS. This is a prospective, multi-center US study in adult participants with ALS who initiate edaravone treatment as a part of their clinical care. Interim results of the study are being presented now at NEALS here in Florida. If you have any questions about any of our products or clinical trials, please feel free to reach out to the email address shown in this slide. I thank you all of you for your attention today.
Thank you very much from Alejandro for that presentation. I guess if I can ask a question, finding valid biomarkers that you raised in that last slide, which I think is an incredibly important point actually. As you said also, it has been quite a difficult thing in ALS research and in ALS studies. Can you share any of the putative that you're looking at or that might have come up in interim analysis from the REFINE ALS data?
We are presenting right now, as I mentioned, the results of this interim analysis. So hopefully soon you're going to have the abstract also available in the website. In terms of the biomarkers, we are assessing oxidative stress biomarkers, 3-nitrotyrosine, protein carbonyls, neuronal injury and death biomarkers like phosphorylated neurofilament heavy and line change, P75 extracellular domain, etc. In terms of muscle injury, creatinine. So there are several biomarkers that we are assessing in this trial.
And when do you anticipate that there'll be kind of some results from that? You might have mentioned this.
Besides the interim results, we hope by next year we're going to have more results.
Okay. Yeah, fantastic. So that really concludes all our presentations for tonight, and we now have some time for questions from the audience. So perhaps we'll open this up to the whole panel. Just to reiterate, we'd love to hear from the audience any of your questions. So if you type that into the Q&A box, and we can read that out for you and put those questions to the panel. And just while those are coming in, I just want to reiterate, thank you again to all the presenters and all the presentations. I think it really showcases a lot of the work that's going into clinical trials in ALS around the world. And I think it's fair to say that this kind of huge increase in activity really reflects the motivation and excitement, I guess, of the MND research community that we're working towards a common goal, and that this will be a condition that can be actively treated. So thanks again for your time and presentations. So to start with, there is one question already in the Q&A, and that's really going to asking about subgroup analyses in the Healy platform trial. Does the Clene group, and particularly the Healy ALS platform trial, anticipate doing any subgroup analysis?
Yes, indeed we do. We are currently waiting for Regimen D to be unblinded. Regimen D is still not announced, and there is no public discussion of the results yet for Regimen D. As soon as that occurs, then the individual companies for Regimen A, B, C, and D, which includes Clene Regimen C, will receive all of the tables, listings, and figures, the entire data set. There are no yet announced for any of the regimens exploratory efficacy measures, and Clene very much looks forward to receiving this entire database from the Harvard Healy team in the coming weeks to do all of these subgroup analyses. Yes.
I guess that's a question that really we can ask of a lot of the clinical trials, given that we've seen in the past that subgroup analyses also can come up with some efficacy that we don't always see in whole group analyses. So I guess that's a question that I don't know if anybody else wants to talk to that from their perspectives. Maybe not particularly. I guess while we're just also waiting for more questions in the chat, one of the things, and we kind of mentioned this earlier on in before we started, but I guess one of the things that is showcased in this presentation tonight is the different types of trial designs that we have, and this debate about the efficacy and robustness of single drug trials versus platform trials. And we've had a mix of those studies today. So it'd be nice to hear about your thoughts around that concept and any tensions about this, and how you really see study designs in the future and going forward, and the general thoughts around that.
Well, perhaps a way to simplify that would be, do you see platform trials as being sufficient as pivotal trials to take forward for approval, or do you think you still need individual phase 3 trials? Obviously, everyone has a vested interest in this, so.
Perhaps I can break the ice on that one. As far as we are concerned, or we're concerned, we were pretty well engaged already in phase 2b/3 programs when some of the platforms trial proposal came. So it was quite quite difficult, I would say, for us to join the platforms in the stage of development we were, also because of regulatory constraints, or at least comments that we got from regulatory agencies around the world. But obviously, probably in my eyes, the most benefits of platforms trials are early in the development rather than late in the development. But it's probably my own opinion and probably not shared by all of us here.
Any other thoughts?
We haven't, this is Steph from Biogen, we haven't participated in the platform trial, but I'll highlight that we're sort of at this, this point in time where it feels like all aspects of clinical trial design are being revisited. And I think we've learned a tremendous amount from many of the trials that have read out in recent memory. And all of the learnings have emphasized the importance of controlling for the heterogeneity that exists and looking at novel endpoints, looking at novel biomarkers, a lot of the things that have been said today. But I think it's an exciting time and it can feel a bit overwhelming, particularly for those that have ongoing pivotal phase 3 studies, because there's only so much that you can do while those studies are ongoing. But I think the trials will look vastly different 5 years from now than they do today. And that's exciting.
Yeah, further on that point, Stephanie, I think Rob alluded to it as well, that perhaps the length of trials has been a bit of a challenge to date, with perhaps the shorter 6-month trials. It seems that nearly all those trials need the OLE or the extended access programs to really tease out the potential beneficial effects of the treatments. And just thinking that with this panel here, do people think that really 12-month trials are probably the way of the future, or longer trials anyway?
I'll break the ice on this one. Clene certainly believes that is the case, especially if one has, as Clene does, a drug that takes a period of time to get to full therapeutic effect. Our asset is 3 to 6 months to full therapeutic effect. We've realized that now from our rescue data. And so our intention is that, like we do already in the MS case, longer studies are critical. Indeed, our MS data was 48 weeks or a year.
Thank you, Rob.
And I guess just, Stephanie. Okay.
I was just going to mention, so when we talk about trial design internally, we often remind ourselves to talk about the control period of the trial, because I think that we can get creative. Many of you may be familiar with the tofersen program, where we had a separate placebo-controlled period and then an open label extension. If we could rewind and do it over again, it would all be under the same protocol. And we would contemplate a crossover design where you're essentially looking at earlier versus delayed start of a therapy. So you could have a shorter period for the placebo-controlled period, and then again looking at that difference of onset of treatment over the longer term. So I think there are a number of different things. I think it depends, as Rob alludes to the timing of onset of each of the individual therapies and the subset of the population that you're looking at. How homogeneous is the decline? How reliable is it that you'll see a decline in those participants over a 6 versus a 12-month period? So there are a lot of factors that come into play.
And I'll just add to that, there are also a number of regulatory considerations that come into play, because Canada and the US accepted a 6-month trial for approvals for our product, but Europe is requiring a 48-week study. So it really is mandated very often by regulatory as well.
From Ferrer, what we are doing is our primary endpoint will be at 48 weeks, but we are continuing a follow-up up to 72 weeks. And then we have the open label extension. So it is, so we're being safe and we're checking all the boxes, but then the regulators are the ones who are actually deciding, and the variability under these decisions, depending on the agencies and the regulators are, well, we will see if these changes as well. We're excited and we want to do different trials and different designs, but the regulators are still, they have their own criteria for the assessing the products.
If I may jump and add on your last point, I think there is a very surprising lack of alignment between regulatory bodies, not only across the Atlantic, but also between what we have been requested, each of us and our companies. I mean, sometimes you see different approaches in time, which are not necessarily linked to the evolving of knowledge in the field, but we see different requirements coming to different companies, different requirements coming from different regulatory authorities. So I think there's also time to probably work towards alignment and harmonization on requirements from regulatory bodies. And I think this is not only the role from pharmaceutical companies or healthcare companies, but also of alliance, of patients associations, and all bodies involved in clinical development.
Yeah, I very much agree. I think it's important that everyone works together towards some common goals, and hopefully those goals are hopefully clear, that where we can all work towards. Clinical trials is just one of those areas. Just a question that probably has direct relevance to myself and Thanuja being in Australia, but we're wondering what other plans to expand the trials into regions. I know some of your trials already are in places like Australia, in the smaller countries, but we're always interested in what can attract you to smaller regions, and also what are the obstacles. And so to give an idea of the local agencies, what they can do to try and encourage trials to come to their regions.
I think there are a number, this is Steph again, I think there are a number of things that go into that the sort of decision as to where to conduct our clinical trials. And there are some local considerations. For example, if you're looking at a biomarker as a key endpoint in your study, and you're measuring said biomarker centrally, conducting a trial in a region like China, where export of samples is a challenge, means that you need to have local testing of that biomarker with the same assays that you're using more globally. So there are some very technical considerations that I think all of us are working to overcome, so that we do have this option. But ultimately, I think exploring conduct of these trials in smaller regions that we haven't conducted as many trials in the past is absolutely essential, because there is a capacity issue. And a lot of the sites that have been conducting all of the studies that many of us are working on are struggling, and you hear it from them directly, that they're struggling to manage all of these different trials. And it's a good problem to have, but I do think that we need to start to explore not only new regions, but also less well-known ALS sites in the regions that we're already conducting studies in.
And I guess slightly related to that, has the pandemic, which is still filtering on in places, impacted any of those decisions, or even your trial design decisions? How has that been of effect?
I can speak to the trial design component. I think incorporating what are, patient-centric, but they're also COVID friendly telemedicine approaches. So like the assessing ALS, FSR, and SVC remotely, and other remote assessments as well. We, from a small molecule perspective, you can do that, but for other, intrathecally delivered products, that's not an option. They still have to go to the clinic.
I can comment on that from Cytokinetics perspective as well. It's certainly a global challenge to be both running clinical trials as well as participating in clinical trials during this pandemic, and remote assessments, trying to have a well-balanced number of both remote versus in-clinic visits to make sure that the trial is run as best as possible, and that you're collecting high quality and good data. So hopefully these will be learnings that we carry forward, and we can all design trials that are the least possible burden to our participants and to the community, while also collecting the best data possible.
And as you say, perhaps this will inform us for the future on how to do this in the most efficient way. Patients always do have trouble sometimes traveling in during those, to centres that are a little bit far away, and that can sometimes impact inclusivity, can't it? So this might actually be the silver lining in that cloud.
Okay, so I think that's all our questions exhaustive today. I'd just like to thank all our presenters again. It's fantastic to be able to hear firsthand from the array of trials and all in one place as well. So hopefully it's really given the ALS MND community a real great update on what's happening and some real hope for the future as well. And we'll all be very keen to see the outcomes of these trials as well. I mean, we're all everyone wants all these trials to succeed, so we have options and treatments for our patients. Thanuja, do you have anything else you want to add before we hand back to Kathy to close off?
I say thank you very much, and yeah, very eagerly awaiting the updates.
Just a huge thank you to everyone for participating today. I know we have some conflicts in people's schedules, and we appreciate the time that you spent here today and updating our community. This webinar will live on our website, so individuals will be able to access it in archive going forward. So very, very much appreciate your time here today, both with the presentations and the questions and answers. Thanks to Dr. Dharmadasa and Dr. Thomas for moderating the webinar as well and bringing this to the global community. And like each of you said in your presentations, we appreciate more than words can say the individuals that participate in clinical trials and work towards a world without ALS. We're grateful for each and every one of you. Thank you, everyone, for your time today.
Thank you.
Thank you.
Thanks again.
Thank you.
You're welcome.
Good night.